Okay, so we talked about the structure of a phospholipid before, right? Okay, so there's 4 parts to the phospholipid. You guys remember what they are? Okay, choline. So I'm not going to draw out the structures, but I'll do different colors. So we got choline on top. Okay, I'm going to use yellow for our phosphate group. Glycogen. How many carbons are in glycogen? Okay, you could do that if you want. Okay, and then the last part. Okay, our fatty acids. All right, which components are hydrophilic? Okay, choline phosphate with glycogen, not so much. Okay, but these are hydrophilic. Which part is hydrophobic? Yeah, especially the fatty acids. Okay, since we're talking about the phospholipid bilayer today, I just thought we should review this. Okay, and how are they arranged in the phospholipid bilayer? Okay, how could you describe it? What's that?
As in like micelles, like how they're structured?
Okay, yeah, but they're not like micelles. They're a little bit different. Right, so they're making a bilayer where the tails are pointed towards each other on both ends, and the heads are towards the outside and inside. Right? So you have heads on one end, heads on this end. And tails pointed towards each other, right? And then goes all the way around, forms a big circle.
And the tails are the fatty acids, right?
Yes, so I switched the colors. The tails are the fatty acids, the hydrophobic regions, attracted toward each other. And then you have a fluid on the outside, which is mostly water, and a fluid on the inside, which is mostly water. And that's the hydrophilic part. Okay, so how well do you think you know the phospholipid structure? Could you erase it and do it again? Okay, why don't you... Okay, so moving on. Cholesterol has a similar structure, not as pronounced as the phospholipids, but cholesterol has a similar structure. It has a little OH on it. And then I'm not going to draw this to scale, but it has a ring structure. And then it has a hydrocarbon tail. Okay, so those are our 3 parts. All right, so how do you think cholesterol would fit next to the phospholipid? Okay, so you should, if you use, let's see if this works. Yeah, that might work. No, I can't move it. Never mind. All right, so where would you draw, if you were to draw the cholesterol next to the phospholipid, how would it line up? How would it arrange? What kind of forces might help bond it within it?
Would its hydrocarbons be opposite?
Yes. Okay, so what kind of forces might hold those together? Van der Waals, good. All right, and then the OH is a polar subgroup. So what force or bond might it form with the hydrophilic end? A hydrogen bond, good. Okay. Either with that or with water, which is going to be on the outside of the cell. So cholesterol fits right into the phospholipid bilayer. It's not as big, it's a little bit smaller. And what it helps to do is break up a little bit of the structure so those tails don't get stuck together. And if there's too many of the, especially the saturated tails in there, it won't allow anything in. So cholesterol helps to increase permeability. And make the structure of the lipid membrane more fluid.
So is the hydrogen bond make it differ between the phosphate or the choline and the OH?
Yeah, just they're polar. So the OH is polar, so it can form a hydrogen bond there. But also the water around it is polar as well, so it can form bonds with the water. Okay, any questions? Other questions? Okay, so we have here a phospholipid bilayer. And I have little components. What's this over here that we've drawn? That's a cholesterol, believe it or not. Okay? And so a cholesterol is going to fit with its little OH coming off the top here. Its ring structure in there. And it's not going to be that big. I'll draw it smaller. Something like that. So it's not doing a lot, but if you get enough cholesterol in there, it just breaks it apart, pushes it a little further apart to allow more substances in there. Okay. Now, the structure itself is called, of the lipid membrane, is called the fluid mosaic model. Okay? It's called fluid because all the components within there don't remain fixed in the same spot. They actually move around a little bit, okay, depending on different forces of attraction and repulsion. And a mosaic means it has a lot of different pieces to it. Okay, so the fluid mosaic model, one of those pieces we already drew in there, that's cholesterol. So you'll have cholesterol spaced out in different parts depending on the different function of the cell to allow for more or less permeability. Okay. And we have other molecules in there. including our integral plasma membrane proteins. So PMP I abbreviated, but that's just plasma membrane protein. Okay. We've got a host of peripheral plasma membrane proteins. And then we have combinations of macromolecules such as glycolipids, and glycoproteins. All right, so I want you to take a minute, and you can erase if you need to. Let's use the bottom one. And talk with your neighbor and discuss where you would draw these different things, what they might be made of, and where they would be located then within the phospholipid bilayer. Glycoprotein has a protein and a sugar on it. And so the glycoprotein might take up a larger chunk, the protein part, across the lipid membrane, and then have the sugar chains coming off the top. Okay, so what I drew here, is this an integral or peripheral protein? Integral, right? So if it's integral if it spans the length of the lipid bilayer. And then if you drew a peripheral one, how would you draw that? All right, so you might have one over here or up over here. And for every cell membrane, you're going to have outside of it the extracellular matrix. And on the inside, you'll have the cytoplasm. And you may have elements of the cytoskeleton. This is getting pretty messy. And the cytoplasm within it. Okay, so this is, again, the fluid mosaic model. You have your phospholipids as your base structure. You have glycolipids, glycoproteins, which are going to be embedded in different areas depending on their attraction to the lipid hydrophobic or hydrophilic part. You have cholesterol arranged also hydrophilic and hydrophobic, which helps break things up and makes it more permeable. Okay, any questions? Cytoskeleton pieces.
And then the cytoplasm is like between the cytoskeleton.
Cytoplasm is just a fluid everywhere. Yeah, so it's all over the place.
Is it like stuff that proteins?
There is. This is a simplified version, but there is a little more detail, which we'll see on the worksheet. Yes. But I don't have any more room, so. Yes?
Does the glycoprotein and the glycolipids, they're not next to each other, you just did that because it's like easy to draw.
Right, yeah. They can be anywhere. They can be, it's fluid. Like they're moving around depending on, their function and where they're needed.
Does this model differ from like
animal cell to a plant cell to
The model is the same, but then the types of proteins are going to be specific to the cell, to the tissue.
but the
To,
basic kind of structure, the things that get added on, that's all the same, just depends on that protein.
yep. Yes. Good questions. All right, any other questions? Okay, so these proteins can have lots of different functions. Okay, and what I thought we could have is an acronym off, where you guys can come up with your own acronyms. I came up with one: teach everything really, really, always classy. Okay. But I want you guys to come up with your own, and then we'll share them and vote on which one is the best. Okay?